Good morning. So in this presentation, we are going to study about the basic methodology and types of the tissue culture. This is the class which is meant for the fourth semester B.Pharm students. And already we have seen about the basic introduction, history, and the requirements for the plant tissue culture method. In continuation of that, now we are going to see the basic methodology and the types of plant tissue culture. In this, we are going to see only the first two types, and the rest of the things we will see in the other presentations. So the basic methodology of the plant tissue culture, it includes the following steps in the process. The first one is the preparation of the suitable nutrient medium as per the selection of the plant medium is autoclaved. That is, in the one of our presentation, we have seen about the media preparations and the substances required for the medium. So for the which kind of cell culture and what type of medium you're going to use, you have to select and you have to weigh the appropriate nutrients required for the preparation, dissolve it in the deionized double distilled water and adjust the pH, add all the required solidifying agents and other things, then you have to autoclave it. Okay, so thereby you have to prepare your suitable nutrient medium. And the second step is the selection of the explant. So already I told you about the explant. It is an any excised part of the healthy plant which is to be used, maybe a bud, leaf, root, or a seed or a shoot or whatever it is. Okay, and once you have selected the explant, it has to be sterilized. So already regarding this also we have seen the various sterilizing agents what you are going to use, and again I'll tell you here, you can use sodium hypochlorite or mercury chloride in the appropriate concentrations, and it is washed aseptically for 6 to 10 times with sterilized water after the sterilizing with the sterilizing agents. And next step is the inoculation of the sterile explant into the solidified nutrient medium under aseptic conditions. And after the inoculation is over, it has to be incubated at 25 degrees centigrade plus or minus 2 at a relative humidity up to 50 to 70% for from 16 hours or the specified period of time. And next step in the basic methodology is regeneration. The plantlets regenerated after transferring a portion of callus into another medium, and induction of roots and shoots or directly from the explants. And next, the hardening. It is the gradual exposure of the plantlets to get accustomed with the environmental conditions. And once the plantlets get hardened, it has to be transferred to the greenhouse and further to the field conditions. So this is the basic methodology where you can see the various steps in this figure. The right from the plant which is in the field, from that the explant is taken, washed well and sterilized, and again washed and trimmed and inoculated into the solid medium, and then it is subcultured once the callus is formed, and after subculturing it starts giving out the buds, and then the microshoots have been formed, and again it is developed into a plantlet along with the roots, then transferred to the greenhouse and then to the open field. Okay, so this is the methodology. It shows the how the plant tissue culture is done, or from an isolated explant how the whole intact plant is developed. And again, this is also another representation. And now we will see about the types of the plant tissue culture. The types of plant tissue culture depending upon the type of the medium used and also the part of the explant what you are using for the culture. So first we will see about the first classification, that is the types of tissue culture method based on the type of medium used. So the tissue culture based on the type of medium, if it is a liquid medium we are using, it is called as your suspension culture. And if it is a static culture, it is called as your Callus culture. And which in turn the liquid culture or suspension culture is divided into the three categories: batch culture or semi-continuous culture or continuous culture. And again, the continuous culture is classified into open type and closed type. Under open type, again, it is classified into turbidostats or chemostats. So now we will see only about this, the types of tissue culture method based on the medium used. So this is the types of tissue culture based on the organ used that we will see in detail in the later presentation. Basically based on the part of the culture used, divided into three. One is the organ culture, that is based upon the different parts of the body what we are using as an explant, like root tip culture, shoot tip culture, anther culture, embryo culture, ovary culture, etc. And the second category is hairy root culture, and the third one is your protoplast or somatic hybridization. And now we'll see the detailed information about the type of tissue culture method based on the medium used. The first one is the static culture or the solid culture. That is the callus. First of all, you should know what do you mean by a callus. It is a mass of undifferentiated plant cells grown on a solid medium from a part plant, which is otherwise called as your explant. This callus may initiate from the explants of any multicellular plant. The organs from which the explant is taken may be from root or stem tips or leaves, flowers, fruits, etc. After the explant is isolated from these different parts of the plant, that can be grown on the solid medium. And the cell groups are initiated from, as I already told you, the explants that may be from the segment of a root, stem, or a leaf, or either from the mature embryogenic plant also. And this explant or the excised fragments may be of parenchymatous cells or mixed tissue containing the cambium or endosperm. When you are taking the explant or isolating the explant from a part of the plant, you have to see, you have to give a great care. The longer the tissue explant, the more complex will be the range of cell types, and there are greater possibilities of initiating the culture of mixed cells. So that is why you have to take or the explant should be as minimum in its size. And this callus can be induced to undergo organogenesis or embryogenesis, and then this can be developed into a whole plant by providing a suitable nutrient medium. That means the growth of the cells either in the callus or in the suspension, it may be by two ways. One is the organogenesis or another one is the embryogenesis. In organogenesis, directly the different parts of the cells are differentiated and grouped and they grow into a whole intact plant. Whereas in case of the embryogenesis, first the embryo is formed, and from that the different organs are initiated to develop into a whole plant. And also this callus is used to study the biosynthetic pathways of various metabolic processes by using the tracer elements, and it is useful for the production of secondary metabolites. So what are the applications of this callus? If you see, you can initiate the organogenesis or embryogenesis, and from that the whole intact plant can be obtained by providing a suitable nutrient medium, or otherwise it is used to study the biosynthetic pathways which is taking place in the various metabolic processes in the cell or a plant by using the tracer techniques. And also it is useful for the production of secondary metabolites. And how will you perform, or how will you do the technique? What is the procedure to do the callus culture? Whatever the operation you are doing, it has to be performed under the aseptic conditions. Whatever, suppose if you are selecting the seed, that seed should be immersed in 70% ethanol for 2 minutes, and it should be rinsed thrice with the distilled water to remove all the alcohol present over it. And then the surface sterilization has to be done. How it is done? By submerging the seeds for 5 minutes in 2% volume by volume bromine solution, or by 2% aqueous solution of sodium hypochlorite. After 5 minutes, it should be washed well with sterile water 3 times or sometimes more to remove the sterilizing agent. And next, we have to inoculate the seeds, or you have to germinate the seeds in the dark for about 2 to 3 days on a sterile filter paper or a cotton wool. And this cotton wool or filter paper, it should be moistened with sterile water, and it has to be kept in the petri dishes. The temperature should be maintained at 26 degrees centigrade plus or minus 2. And once the germination has come out from the seeds, the cotyledons are removed carefully by cutting with the sterile scalpel, and transfer the explant portion onto a solid sterile medium in the cultured flask. And once the cotyledons, the explant is cultured, it is incubated at 26 degrees centigrade plus or minus 1 degree in the darkness for about 3 weeks. And it should be observed for the growth of the callus. And transfer the culture aseptically on to the sterile fresh medium at the interval of 4 weeks. And once it is grown, the growth index can be calculated by using the formula final weight of the callus by the initial weight of the callus. Usually this static culture method is used to detect the plant metabolites and to study the precursors used in the bioproduction of secondary metabolites. So this is the procedure to perform or to develop a callus culture. And next one is the suspension culture. It is an example coming under the medium or the nutrient medium in the liquid condition. Suspension culture involves the active proliferation of callus as undivided unit suspended or submerged into the liquid medium. The nutrient medium is in constant agitation so as to prevent the cells from settling or aggregating into clumps. Suspension cultures are initiated by transferring the pieces of undifferentiated callus into a liquid medium which is agitated during the incubation. So this suspension culture is again in turn divided into a batch culture and the continuous culture. So what do you mean by a batch culture? This means the culture in a fixed volume of culture medium. It is a closed system where the specified amount of the culture medium is taken and it is inoculated, and once the cells are grown fully or once the medium is deprived of its nutrients, the process is stopped and then it is subcultured. Or suppose if it is meant for the production of secondary metabolites, the secondary metabolites can be removed by filtration and then the fresh subculturing can be done. In general, the nutrient medium or cellular inoculum are mixed and they are aerated and allowed to grow in a fixed volume of culture medium. In the batch culture, as the cells grow, the medium is depleted of nutrients, as I already told you, and the metabolic byproducts and the cells gets accumulated. So this is the time you have to stop the process and remove all the metabolites or the cells, and you have to reculture it. Batch cultures are characterized by continuous changes in the medium and continuous internal changes in the cellular composition, or otherwise the accumulation of metabolic products. So these are the three ways by which you can characterize the batch cultures. The batch culture is a closed system with respect to the additions or removal of cultures except for circulation of air. That means what? Once for all, the medium is taken, it is inoculated, allowed for a specific period of time for the process to run, and once the changes has been happened continuously, it is stopped, the subculturing is done. The next type in the suspension culture is semi-continuous suspension culture. Here the system may be open, and here what happens, periodically the culture can be removed and the fresh medium can be added into it, and the growth of the culture is continuously maintained. And next one is the continuous suspension culture. Here also this is a open system of your suspension culture, and the system volume, in this system the volume of the culture remains constant. How it will remain constant? Automatically the fresh medium is continuously added into a bioreactor so that the fixed volume of the culture remains the same throughout the period, and once the growth is attaining its maximum growth, it can be withdrawn automatically and continuously without stopping the process. Whereas in case of your batch culture, what you are doing, you are adding a fixed volume of a culture media, and also you are inoculating, allowing the specified time period to grow or the production of the metabolites, secondary metabolites. Once it is over, you are stopping the process and collecting the grown-up cells or the secondary metabolites, whatever it has been produced, and the fresh culture media is added in case of the batch culture. Whereas in case of your continuous suspension culture, you are not doing it so, because it is a continuous culture. So here it is, all the things are predetermined. The culture medium is continuously allowed to flow into the bioreactor, and the cells are inoculated. It is allowed to grow, or it is allowed to produce the secondary metabolites. And once the growth is over or the secondary metabolite is produced, it is automatically removed from the bioreactor by the proper adjustments, and continuously the fresh culture medium is fed into the bioreactor without stopping the process. So in this continuous cell suspension culture, the cell proliferation takes place under constant condition. That means there is no variation. Why? Because you keep on adding the nutrient medium or the culture medium, and also simultaneously you are removing the cells grown up or the secondary metabolites produced. So always the cell proliferation as well as the production takes place under the constant condition. This system allows the establishment of steady states of growth as well as your metabolism of the cells. That is, in other words, the production of the secondary metabolites. The study changes which occurs in transitions from one steady state to another state. And identification of the controlling factors also can be monitored over here. And there are two types of suspension culture. One is the open type. In that is called as your chemostat, and the other one is your turbidostat. Chemostat is nothing but, that means it is a continuous new medium input is set at a predetermined rate and determine the nature of the resulting equilibrium. That's what I told you already. The inflow of the culture medium or the nutrient medium is predetermined, and it is allowed to flow continuously into the bioreactor. And once the growth of the cell is attained, there will be a change in the chemical nature of the culture medium. That means what? Maybe whatever the oxygen which is provided inside might have got used by the cells, and more amount of carbon dioxide may be produced, or the other unwanted waste materials produced by the cells which is present in the bioreactor may result in changes of the chemical nature of the culture medium. So that will be sensed by the sensors, and automatically the fresh culture medium is allowed to let inside, and whatever it has been produced earlier in the bioreactor will be automatically removed. Okay. So that is why the desired rate of growth is maintained by adjusting the level of concentration of nutrient by constant inflow of the medium. I think you got my point. There will be sensors which is fixed in the bioreactor, which will sense the change in the chemical nature of the medium, which is resulting due to the complete growth of the cell or by the complete production of the secondary metabolites. Once this chemical change is sensed, automatically the new or the fresh culture medium is allowed to flow into the bioreactor by the predetermined regulators which is fixed on it. And next one is turbidostat. This is nothing but, here the turbidity of the culture medium can be determined And once there is a increase in the turbidity, this allows the inflow of the fresh medium into the bioreactor and the removal of the cultured cells from the bioreactor. The cell density is set at a predetermined level and new medium is added to maintain the cell density within the limits. Okay, so here that means when the medium is added and the cell is allowed to grow, when there is a complete growth of the cells is happening over there, there will be the difference in the turbidity of the culture medium. So when the turbidity increases, the sensors will sense the increase in the turbidity and automatically the fresh nutrient medium will be allowed to flow into the bioreactor. Okay, so that is the continuous suspension culture, that is the turbidostat. And this method is valuable for work at a low concentrations always. And it is a simplified system where we can study the effects of physical factors, growth regulating substances in growth rate, and their associated metabolism can be studied by this method. And next one is the closed type of your suspension culture. In this, the used medium is replaced with the fresh medium, and the cells from the used medium are mechanically separated and added back to the culture, and thus the cell mass keeps increasing. This is the closed type of your suspension culture. And how this suspension culture technique is done. The suspension medium is taken in a conical flask and it is autoclaved, and the sterile medium should be used for the technique. And already from the callus, the pre-established callus, specified weight should be taken and it should be introduced into the conical flask, and the conical flask should be closed with a cotton plug and it is placed in the clamps of the rotatory shaker, which moves at the speed of 8 to 120 rotations per minute. So whatever the transferring or the aseptic things, whatever you are doing, everything should be done in an aseptic manner. And after considerable time, that is around 3 to 7 days, the entire contents of the flask has to be taken. It has to be filtered through the sterilized sieve, and the collected filtrate is collected in a pre-sterilized container. And this filtrate is again centrifuged, the supernatant is poured off, and the residue is the free cells or the cell aggregates. And these cells are again cultured into a fresh liquid medium, and the flasks are again agitated by a shaker so that each and every individual cells are suspended equally in the liquid culture medium in the flask. So by this way, the same process is repeated to suspend each and every individual cell in a nutrient medium or the culture medium. So from this culture, the inoculum is subcultured in a pre-sterilized liquid medium at regular intervals, and it has to be maintained. And these cell aggregates are again and again, it is subcultured and it is kept in the culture room for the various studies like regeneration of the whole intact plant or for the production of the secondary metabolites. So that is the procedure or the technique you will use to develop the suspension culture from the callus. And now we have to see the growth characteristics of the suspension culture. The growth characters can be studied, or the cultivation of the cells in the suspension culture or the media culture is by two ways. One is the organogenesis and another one is the embryogenesis. So what do you mean by this organogenesis? Organogenesis is a process. This involves the redifferentiation of meristematic cells present in the callus into the formation of shoots or buds or roots or even the whole intact plant. That means what? From the growing cells which is present in the callus is differentiated into different parts or organs of the plant like shoot, bud, root, or even into a whole plant. That is called as organogenesis. That is formation of each and every organ of a plant from the growing cell present in a callus. Understood? That is called as your organogenesis. In the other way, the formation of organs is called organogenesis. What are the organs? It may be a different part of the plant like your shoot, bud, root, etc. Organogenesis is a process which is very much unique to each and every plant. In the plants, if you take the development, it is the process which results in the functional maturity of the organ, which includes all the events in the life of the plant to produce the various organs of the plant. Okay, that is in the plant development, you can say that the formation of each and every organ, the functional organ in the body of the plant is one criteria. And another criteria, what happens, that is the provision of capacity to obtain the food or to reproduce or to deal with the hazards of its environment. That means, in other ways, to protect itself from the environment. So these 2 points are very, very essential in a plant development. That is, what do you mean by a plant development? The first point is development of the different organs of the plant. And second one is the maintenance of its physiological functions of each and every organ. That is the developmental process in the plant. The first one is forming up of the anatomical organs of the plant, and second one is the maintenance of the physiological functions of that organs. That is called as the plant development. In the plant development, these are the 2 processes. One is the formation of the different types of the organs, and next one is the functions of these organs maintenance. Understand? Yes. And then the shoot buds or the monopolar structures, which is giving rise to the formation of the leaf primordia or the apical meristem. From the callus, the very first part formed is your shoot, from where the roots and the other parts of the plant like your leaf primordia or apical meristems are formed. The stimulation of shoot bud differentiation depends upon many factors, and this is also different in each and every plant species. That is, the formation of shoot bud in the culture depends upon many factors, and from plant to plant it will vary. So this was demonstrated by a scientist called Skoog, and he told that the auxins, the plant growth hormone, will stimulate the root formation in the shoots, and in the other way, it will inhibit the shoot formation. And apart from this, there are other factors which affects the organogenesis are the size and source of the explant. That means what is the size you are taking. Already I told you, if your explant is bigger, there will be more complications. So the size of the explant what you are taking should be of minimal size. And also the source of the plant. The plant should be well matured and healthy enough to have totipotency. That means totipotency is nothing but it is a capacity of an individual cell to develop into a whole intact plant. Okay, so all these things will affect the organogenesis. And apart from this, the light intensity also plays an important role in organogenesis. So how does this light intensity affect? If there is a high light intensity, it shows the inhibitory effect on shoot bud formation in case of tobacco plants. And apart from the light intensity, the quality of the light also has the effect. Here, if you take the blue light, it induces the shoot formation, whereas the red light stimulates rooting in the tobacco. And apart from the light, the temperature also is important. We have to provide the optimum temperature for the explant to initiate into the different parts of the plant or different organs. And usually the solidified media with the agar favors the bud formation, and in some cases the suspension culture favors the formation of organogenesis. And next is the embryogenesis. What do you mean by embryogenesis? What do you mean by an embryo? Usually if you say an embryo, where it for the formation of embryo, it needs to have the fertilization. That is, the male gamete and the female gamete should fuse together, and then it should form a zygote, and from the zygote the embryo is formed. But whereas in the organogenesis, from the part of a plant, the different organs are differentiated from the undifferentiated callus. So now we will see embryogenesis in detail. Embryo is defined as the multicellular stage of an individual, which occurs before the development of characteristic organs in each and every species. The production of embryo from a callus is known as embryogenesis. When you see in higher plants, the embryo arise usually from the zygote, as I already told you, and they are termed as zygotic embryos. And if you see the sources of embryogenic cells, they may be of microspores, that is 1N, you can call it as. And zygote, it has 2N configuration, and somatic cells as well 2N, and the somatic hybrids will have 4N. This embryogenesis, it can induce, or embryogenesis can be induced by non-proliferative or proliferative direct embryogenesis. And the growing of embryogenic callus, which in turn germinates and develops into a whole plant. Okay, so now I think you will get the difference between the organogenesis and embryogenesis. In the organogenesis, from the explant, from a direct part of a plant, it is differentiated into different organs by the cell proliferation. But here, the embryogenesis, the formation of embryo is initiated first from the non-proliferative or proliferative embryogenesis. And from that embryo, the cells gets germinated, the callus germinates, and it is developed into a whole plant. This embryogenesis can be initiated in an explant from the juvenile or meristematic tissue. What do you mean by juvenile? That is from the young plants. Meristematic means growing tissues. Okay. And immature zygotic embryos, that is the examples may be the immature zygotic embryos, that the explants, the examples of explants, sometimes the cotyledons, which is present in the seed, the embryo, or the hypocotyles, which is dissected from the ungerminated seeds. These are all the common zygotic explants, or the embryogenesis can be initiated by the explant from the cotyledons or hypocotyle dissected from ungerminated seeds, or sometimes from the immature zygotic embryos. Isolated somatic cells can also develop into an embryo sometimes. This embryo development occurs through an organized sequence of cell division followed by cell enlargement and cell differentiation. The final stage of development towards the maturation or characterized by overall enlargement or matured embryo morphology. Okay, so here the organs or it is the whole intact plant is developed from the zygote or the embryos. Somatic embryogenesis involves three distinct steps, which is not we can see in the organogenesis, or which is not found in the organogenesis. So what are that three distinct steps involved in somatic embryogenesis are the induction, maturation, and conversion. What do you mean by that induction? What is happening in induction? So it is an initiative phase where the callus cells are induced to divide and differentiate into groups of meristematic cells called as embryogenic clumps. Okay, that is in induction. The embryogenic clumps are developed from the callus. So how it is developed from the callus? The cells from the callus divide, and then they differentiate into group of growing parts of the cells. That is otherwise called as meristematic cells. That is called as your embryogenic clump. And these embryogenic clumps develop into the initial stages of somatic embryo. That means initially they develop into a stage which is called as your globular stage. That is the initial stage. And next one is the maturation. The somatic embryos which are developed as globular stage will mature into embryos. That is how they will be maturing. The initial globular stage is converted into heart-shaped and mature embryos, and they undergo biochemical changes and acquire hardness. That is called as your maturation. Understood? The first step in somatic embryogenesis is induction. In this induction, what happens? From the cells of the callus, it divided into many number of cells and differentiate into group of meristematic cells, and this differentiated cells are called as embryogenic clumps. And this embryogenic clumps in the initial stage, they are called as the somatic embryo, that is otherwise called as globular stage. And next it enters into the second stage called as maturation. Here what happens? The initial globular stage, the cells are globular in nature and they are developed into heart-shaped cells and they mature into embryo by undergoing biochemical changes and they acquire hardness. Okay, so here what happens in the maturation, the globular stage is converted into a heart-shaped cells and undergo many of the biochemical changes and they become hardened in nature so that it get attempts to grow in a next stage. That is called as your maturation. And next one is the conversion. In this stage, the embryos germinate to produce seedlings. Understand? So these are the three different stages in somatic embryogenesis, where this is not happening in organogenesis. Okay, so what are the three different stages in somatic embryogenesis? Induction, maturation, conversion. In induction, what happens? The embryogenic clumps are induced from the callus, and in the maturation, the conversion of globular stage into heart-shaped cells by biochemical changes and it becomes hardened. And in the conversion, the embryos germinate to produce seedlings. Clear? And next we have to see about the advantages of organogenesis and embryogenesis. So what are the advantages? First one is the efficiency of the process. That means reduction in the labor cost, reduction in time, and formation of plantlets is within the fewer steps. And next one, it is highly potential for the production of um, the plants in much higher number and the cytologically or morphologically uh, uniformity can be maintained in the production of plantlets. And apart from that, the several plants can be produced on commercial scale, which can be uh, useful in the production of food crops or vegetable species or fruits, and also the production of plants with high medicinal values, and also the aromatic plants also can be produced by this organogenesis or embryogenesis. Okay, the advantages, if you see, first one, it is highly efficient by reducing the time, the labor cost, okay, and also the formation of new plant or the plantlets in a fewer steps. And next one, there is a high potential for the production of the plants in more number, and also the similar plants, with the plants which are produced are similar morphologically and cytologically, and also the uniformity in the production of plantlets can be obtained here. And the next advantage what you can see is production of several plants on the commercial scale. That means the plants which are useful as a food crops or the as a vegetables or as spices or as fruits or as a medicinal plants or as an aromatic plants, more number of plants can be produced in a shorter time with uniformity in quality and quantity. Okay, so these are all the advantages of organogenesis and embryogenesis. So that is about the discussion on the types of plant tissue culture based on the culture media. Okay, the plant tissue culture methods can be divided into two types majorly, that is static culture and suspension culture. Static culture is nothing but growing the cells on the solid media, that is nothing but your callus culture. Okay, you have seen what are the callus and how it is developed. And next one is the suspension culture. It is again divided into the many types, that is batch culture and continuous culture. And in the continuous culture, again you have seen about the uh, turbidostats, that is open type and the closed type. And in the open type, again, you have seen about the turbidostats and the chemostats. And then you have seen about the technique for the development of the suspension culture and how the cells or the growth characteristics are obtained in this suspension culture or callus culture has been studied. That is nothing but the organogenesis or embryogenesis. Clear? And also you have seen about the advantages of organogenesis and embryogenesis. So that is about the uh, type of tissue culture based on culture media. Thank you.